Good morning, everyone. Welcome to Medicine Grand Rounds. I'm Abraham Verghese, the Vice Chair for Medicine. I used to host Grand Rounds when it was live, but when we made that pivot to Zoom, we were so delighted to have Errol Osdalga show us the way, our communication expert. We are just so proud and in awe of him. But he's human and needs a vacation, so he's taking a break somewhere near Monterey today, and he will be back next week. So before we start the formal proceedings, I'm going to ask Dr. Neera Ahuja from Hospital Medicine to give us an update, please.
Thank you, Abraham. Good morning, everyone. I'll share my screen and want to start with just the general COVID updates. You know, I wanted to share this slide, which I thought was compelling as we take kind of a global view. Israel is now the #1 country in number of new cases per population, and you can see the US right here with numbers slightly coming down as depicted in this graph. So happy to see the numbers coming down, but you know, they still are a substantial amount compared to many of the other countries around the globe. Even looking within California, you know, we had our big peak in July and numbers coming down, and we'll take a look in the next week or so to see if there is a post-Labor Day effect as we've seen from other major holidays. This morning, we have 19 COVID patients in the hospital, of which 4 are in the ICU and 3 are intubated. We've had 409 COVID patients admitted for symptoms within SHC. Just focusing on the SHC numbers, we still stay around 20-ish% for our ICU rates, and the death rate is about 6.4%. We've had just slightly over 50% females, and once again, that includes, you know, the pregnant women that are in this bunch, so they don't get admitted to LPCH if they have COVID. They do come to our SHC hospital. And here you can see the age distribution of the patients affected, which has stayed about the same over the past 2 months. And then finally, just taking a look at the breakdown of the death, both by gender and age. You know, the majority of the deaths have occurred in the older population, and we'll continue to keep an eye on these trends. Thanks to my data team here, and I will turn it back over to you, Abraham. Thank you.
Thank you so much, Neera, for that wonderful update, and kudos to the hospital medicine team for all the hard work you're doing. I should mention that yesterday we reached a milestone in ambulatory medicine of 100,000 ambulatory tests in the drive-in and other areas. So kudos to our primary care and ambulatory medicine teams who are also doing an outstanding job. Before I introduce the speaker, just a quick mention about upcoming Grand Rounds. I'll share my screen. As you can see, next week we have an approach to monoclonal antibodies, and that'll be from David Weinrich, Senior Vice President at Regeneron. The following week, it's Robert Califf, former FDA Commissioner. And then on September 30th, it's wearables and early detection of COVID-19 using smartwatch by Mike Snyder and I think that's meant to be Steve Asherman. But now it's my great pleasure to introduce our speaker today, leading the panel, and she will introduce the other panelists. And our speaker today is Kari Nadeau. She is the Naddisy Foundation Endowed Professor of Medicine and Pediatrics. And for more than 30 years, she's really devoted herself to understanding how environmental and genetic factors affect the risk of developing allergies and asthma, and laying out the molecular mechanisms for the same. She really is one of the nation's foremost experts in allergy and immunology, leading to her involvement in vaccines, as you will hear today. She received her MD and PhD from Harvard Medical School, did her residency at Boston Children's Hospital, and her allergy immunology fellowship here with us at Stanford. She's on multiple editorial boards, many kudos from different awards from different organizations, publications, patents. I don't want to eat into her time. I should just mention in closing that she's also passionate about breaking down healthcare barriers, ensuring racial equity, very involved in underserved areas in Palo Alto, Harlem, inner city Chicago, and her work has really directly benefited my own center presence. So we're very grateful to have Kari take over now and introduce the other panelists in time. We don't hear you, Kari. Can you unmute? There you go.
Thank you so much, Dr. Vikings. It's a pleasure to be here today. It was very exciting for me to be invited to give a vaccine update, and importantly, I will be sharing this presentation with my colleagues, Dr. Barry and Dr. Grant, to be able to give you last up-to-date minute updates for the vaccine development for COVID. Our outline is the following. I'll still quickly talk about the development timeline of the vaccine, then move forward with vaccine platforms and specific examples in human trials, then move forward with global distribution and considerations by Dr. Barry. Dr. Grant will then give Stanford updates specifically on clinical trials for vaccines occurring here at Stanford, and then I'll end with a resource summary. So when you think about the typical timeline for the traditional development of vaccines, it's usually about 10 or plus years. The attrition rates that are reported are greater than 90%. The fastest development ever is about 4 years for the first mumps vaccine, but that actually started back in World War II, so it's probably a little bit longer than that. Importantly for COVID-19, many people around the country are trying to target a timeline that's more in the 12 to 18 month range. And to do that, there's been some fundamental step changes that needed to occur. Number one, there's some novel platform technologies, which we'll talk about today, including RNA and DNA viruses. There's expedited and innovative regulatory processes now in play with the FDA, and we'll talk about the Emergency Use Authorization Act as well. There are parallel processes occurring with phase 1 trials with non-clinical research, and there's adaptive clinical phase trials that are occurring now, as well as innovative ways to manufacture the vaccine. Importantly is there are increased financial risks with this, and what we'll talk about today is what are the roles of the EUA and the Operation Warp Speed, as well as what should our expectations be, and then finally, what are the challenges for vaccine development in this fast timeline? So when we think about the EUA, it is an authorization act that's been around for years. It's not been used in healthy controls before, and it has not been used for vaccines before. In June 2020, the FDA, CBER in specific, gave advice on some warnings around using the EUA for vaccine development. And they warned that the issuance of an EUA-based action is very carefully and very cautiously based on preliminary safety and efficacy data for randomized controlled studies. And it does reduce the ability to demonstrate effectiveness and assess benefits versus the risk of the vaccine to support licensure. And importantly, the EUA would be made on a case-by-case basis, considering target populations, characteristics of the product, preclinical and human clinical study data, and the totality of available relevant scientific evidence. And for this reason, many companies now have suggested that are making COVID vaccines that they would prefer not to move forward with the EUA Act unless large phase 3 clinical data are available that show safety and efficacy. One of the other ways to hasten development of a vaccine is Operation Warp Speed. And this again is not a new concept. This actually started with the polio vaccine for the Salk and Sabin vaccine, which were manufactured very early, even before their phase 3 equivalent efficacy data and safety data were available. And with Operation Warp Speed, this aims to deliver 300 million vaccine doses in early 2021 as part of a broader strategy. There's a partnership with Human Health and Services, with CDC, with FDA, with NIH, with BARDA, and the Department of Defense. Congress directed almost $10 billion to this effort, and over 6.5 was designated through BARDA, and $3 billion was designated through the NIH. And I just list there the different vaccine studies that have been included in Operation Warp Speed. With that in mind, I want to bring us back to the developmental timeline. You can see here in a nice review that was done in the New England Journal that typically the different phases of the studies are done, and then once you have licensure, then typically large-scale manufacturing then ensues. However, what we're looking at for the outbreak paradigm with overlapping phases is that manufacturing starts very early, even when a target ID is identified, and then once licensure occurs, then large-scale manufacturing then moves forward quickly. So what should we expect at this finish line? And importantly, I'd like to highlight those notable quotes from people that are involved in this area. Dr. Tony Fauci, the NIAID director, warns that the chances of scientists creating a highly effective vaccine, one that provides 98% or more guaranteed protection for the virus, are slim. Scientists are hoping for a coronavirus vaccine that's at least 75% effective, but 50% or 60% effective would be acceptable too. And you've got to think of the vaccine as a tool to be able to get the pandemic to no longer be a pandemic, but something that's just well controlled. Mike Ryan of the WHO, in being cautious about the data so far that we're seeing, which I'll talk about today in phase 2 trials, that really a phase 3 trial means this is the first time this vaccine has been put into the general population into otherwise healthy individuals to see if the vaccine was effective against natural infections. So again, really making sure that we know that the first one out of the gate in terms of vaccine development is not necessarily the one to cross the finish line. And importantly, that we need phase 3 data to be able to generalize to the global population. And then finally, the FDA commissioner has said that it will authorize a vaccine if 50% effectiveness is obtained. So what are some of the challenges that we face now to be able to get to these timelines? First, stability in administration is very important. The ideal candidate should be thermostable, should be self-administered, ideally. It's dependent upon the quantity that's also required. We have the ability to understand how many doses are required per vaccine and what will be required to protect an individual. There are distribution challenges, including limited quantities and who will get vaccinated first and in what order. How will we deal with the shortfall potentially of healthcare workers? And then importantly, what are some of the safety and efficacy issues in this political climate? The ethics of placebo-controlled trials in vaccine studies is also important to be able to examine. And then finally, ethics of access and distribution, especially to those in need, like the underserved populations, the populations at high risk, and the elderly. Other challenges include what type of immune parameters are we going to expect of this vaccine? It's important to note that many of these biomarkers are now being used as surrogates to be able to understand whether or not a vaccine is working or not. And past research on SARS and MERS provides some potential starting points, and you'll hear that from our colleague next week from Regeneron. Importantly, there's an ongoing debate as to what the best target for a vaccine is. SARS-CoV-2 is a large virus that harbors many different types of structural proteins. You'll hear a lot about that spike protein today, the envelope, membrane, as well as nucleocapsid. There's 16 non-structural proteins that also might be important for immunity. And then there's other issues in terms of mutation rate and variants within the virus itself. The correlation of protection has not been established. We need to really understand natural immunity and whether or not that protects against reinfection to know if we are understanding the role of vaccines to be able to emulate what a natural immune system would look like. Today, I'll show you data about neutralizing antibodies and how those potentially could be protective of disease. But importantly, the assays for any of these are critical to be able to assess their relevance. When we talk about neutralizing antibodies, oftentimes a pseudoviral assay is used, and that can be highly variable depending on the company and depending on what kit is used. Importantly, we need to understand the role of B cells and antibodies, but not only the quantity of antibodies, but also the function of them, and those relate to neutralizing antibody studies. Finally, T cells play a role. This has been found by our colleagues around the country as well as by people at Stanford. There's a certain T cell that makes interferon gamma, which we'll show you examples of today, and that's called the Th1 cell. And then innate immunity also plays a role. The candidates right now mostly focus on spike protein and the receptor binding domain, and that recognizes the ACE2 receptor, which is one of the ways the virus gets into our body, so people are focusing on that. Developmental landscape right now looks like the following. This is from the New York Times updates on these vaccines, which you can get as a resource. I'll show you that at the end of my talk today. But importantly is that you can see there's limited approvals already for 3 vaccines in the world, and 2 of them are adenoviral vector vaccines and 1 is an inactivated vaccine. Currently, there are no vaccines approved for full use. The majority are designed to neutralize antibodies against the S protein, and they have different types of platforms. So let's talk about those platforms, because you're probably reading in the news about different types of virus platforms, and they include inactivated viruses. We know this from a lot of our vaccines that we use currently. These are an established precedent in this type of platform. They do have stability. You do need to add an adjuvant to be able to increase immunogenicity. The production speed, though, however, is long because you need to inactivate the virus, and it is scalable because there are already infrastructures and resources that are available to be able to produce these types of vaccines. A non-replicating viral platform, otherwise seen currently with adenoviral vectors, use the adenovirus sort of as a way to co-opt into the shell of a virus and use a transgene expression, for example, of the S protein, to be able to create a viral vaccine that is stable, that is highly immunogenic, and unfortunately can lead to overly strong immune responses. The production speed is about medium in nature. It is scalable, again, because of outstanding infrastructure we have already available. And then there has been established precedent for the use of these types of vaccines. RNA vaccines, the stability is variable. People are working on improving that stability, even here at Stanford. The typical immune response is currently in research phase now. I'll talk about some of that data. The production speed can be quick, but the scalable production is questionable, low to medium, and there is no precedent right now for this type of vaccine. For DNA vaccines, the stability is also variable. It's in research phase, could be done quickly in terms of production, but again, scalability is currently in question and there's no precedence. The protein subunit vaccines also have a variable stability. However, that could be modified given different stability factors within a vaccine product. Typically, you need to add an adjuvant. We'll talk about that today with the Novavax vaccine. And it does tend to be fast, but it depends on the cell line that makes the protein subunit, and it is scalable, and there is available precedent. I wanted to give you a summary of what's out there now in terms of phase 1, 2, and 3 studies for vaccine development in humans. And you can see the majority of these across represent many of the vaccine types that we've just spoken about, and that some of them include 1 single dose versus 2 doses. 2 doses vary between 0 to 14 days, 0 to 21, 0 to 28. Some of them actually include 3 doses, but this is still in research phase. And the arrows represent those vaccines that we are going to be using at Stanford hopefully soon, and Dr. Grant will be speaking about this. I wanted to give you some examples of these types of platforms that are currently in human studies or have already gotten limited approval. This is the inactivated SARS-CoV-2 made by Sinovac and CoronaVac in China. They had some preliminary strong data in Science back in May from rhesus macaque models, and now they've done a phase 2 trial showing a good immune response in over 90% of subjects, and this was obtained after 14 days. They're currently running a phase 3 trial in Brazil. And I'd like to now focus on an adenoviral vector example. This was a vaccine made by University of Oxford and Astrazeneca. It was published in The Lancet recently by Foligati et al. This was their phase 1/2 randomized study results. They are currently enrolling elderly and pediatric patients. As of yesterday, it is on hold. There was 1 case of transverse myelitis that will need further investigations. Many vaccines, by the way, have transverse myelitis cases associated with them, for example, hepatitis, as well as MMR vaccine. But importantly, when any of this happens, it's very critical to look at the data and assess the robustness of the immune response and if this is an autoimmune type reaction in nature. And again, this is an adenoviral 5 vector. That's a different type of adenovirus. It was meant to be mostly tropic for chimpanzees, but there are humans that have a common cold associated with adenoviral vector. So it's possible that there are people that have immune responses already to these adenoviruses that are being used as co-opting devices for the spike protein transgenes that are being produced in this virus. And when you look at immunoglobulin responses to the SARS-CoV-2 spike protein, in the y-axis, you can see the ELISA units, or the immunoglobulin units. On the x-axis of these graphs, you can see the days since vaccination. In this trial, for example, there were 543 people that got active, 534 that got placebo. This was a single dose study for the majority of patients. 10, however, got a boost at 28 days. And you can see that in the third panel. When you look at this data compared to the convalescent plasma samples, it looks like people are getting up to the 10 to the 4th range in their antibody production. Now, I want to make sure that we're cautious in the fact that we assume that the convalescent plasma samples have an immunoglobulin titer that will protect someone against a reinfection, but we don't know that for sure. So we're going off assumptions that immunoglobulin amounts in convalescent plasma samples from already convalescing patients with COVID would be protective, but we don't know that yet either. So what about neutralizing antibodies? This is again the same adenoviral vector Oxford Astrazeneca product that was done in phase 1/2 trials published in the Lancet. And you can see in their patients that overall, with one single dose of vaccine, that you can get neutralizing antibodies in the median and interquartile ranges similar to convalescent plasma neutralizing antibodies. So now I'd like to move forward with the Pfizer product. This represents a nanoparticle mRNA vaccine. Again, these RNA vaccines have not been used before, but there's a lot of hope and promise around these. This is a global phase 2/3 that's currently in 30,000 participants. The findings here are preprints using one of their products. They're moving forward with another product in phase 2/3. The doses here were on dose day 0 and day 28. And again, you can see in the y-axis the immunoglobulin titers to the receptor binding protein or the spike protein part of the SARS-CoV-2 virus, and very similar to what they're seeing in their convalescent serum, which is over on the right-hand side. When we look at neutralizing antibodies, there was a strong correlation between the neutralizing antibodies amount and the titers to the immunoglobulin in these particular patients. But I wanted to emphasize this particular trial because they actually studied how well the immune system made antibodies to the variants of the SARS-CoV-2 virus. And this might be important in the future as we look at potential mutation rates of the virus. Another RNA vaccine which has received a lot of attention in the media is the Moderna mRNA vaccine, also co-sponsored and developed with the NIAID. This is a paper published by Jackson et al., in which 45 patients were given active, but there was no placebo group here. They were dosed at day 0 and day 28. And you can see again in the left-hand panel, similarly, they have titers in the 10 to the 4th area of the y-axis, and that's similar to their convalescent plasma, which they compared against. And there does seem to be maybe a trend for increased titers with increased doses, 25 mics versus 100 versus 250. On the right-hand panel are their neutralizing antibody titers. Important is to look at safety with all of these virus vaccines. We talked about how important it is to look at both safety and efficacy. This is the preliminary safety data. After 2 doses, you can see that many of the adverse events occurred in 100% of the population. These definitions were MEDRA definitions, which are standard definition used when talking about adverse event in vaccine trials. And I'd like to point out over on the right-hand side, as we spoke about, T-cell responses are also very important to be able to evaluate. And here in day 1, day 28, and day 43, after a single dose, as well as another repeated dose at day 28 of the vaccine, there was an increase in the Th1 response and interferon gamma and other mediators of T cell responses. Finally, I'd like to end with a vaccine that represents the platform using a protein subunit with an adjuvant, which was saponin in this particular example. This was just published last week in the New England Journal. This uses a recombinant nanoparticle with a full-length transmembrane domain of the spike protein. This is dosed at day 0 and day 21. 83 people got the vaccine with adjuvant, 25 vaccines were given without the adjuvant, and then there were 23 placebo patients in the study. You can see again in the y-axis, the titers they're seeing are about 10 to the 4 to 10 to the 5th range. This compares to their human convalescent serum controls. And with the additive effect of the adjuvant, you can see there was an increase after day 21 with the second dose of the vaccine compared to no adjuvant. Similarly, it's important to look at safety in the same MEDRA definitions. Looking at the safety here, less people got adverse events compared to the prior mRNA vaccine we spoke about with Moderna, but it's important not to compare these trials against each other. These are separate trials, these are preliminary data, and this is the safety profile so far seen with the Novavax vaccine. I'd also like to point out their Th1 results after their 2 doses. They did see an increase by day 28 in the Th1 response. And we've talked mostly about vaccines that were given intramuscularly as well as subcutaneously, but I did want to talk just at least to let you know that there are oral vaccines being moved forward, although these are much earlier stage. So I'll stop there and introduce my colleague, Dr. Berry, who will speak about the global distribution and considerations for vaccines. Dr. Berry, I very much appreciate you being here, and she's our Dean for Global Health and also is an infectious disease professor. Dr. Berry, I'll run your slides for you. Thank you.
Kari, thank you very much. Kari asked if I would talk about the global distribution of vaccines. And it's really been a global scramble with quite a few government deals with pharmaceuticals. It's led to already a multi-billion-dollar vaccine industry. But when you think about this, it could lead to inequitable distribution, accelerated vaccine development without safety assurances, and also generate geopolitical tensions. So what can we do to prevent that? I think the WHO has been very active, as well as the Coalition for Epidemic Preparedness and Innovations, and Gavi, the Global Alliance for Vaccine and Immunizations. The result of this global cooperative approach has been what people are calling the COVAX pillar and Gavi AMC. And what I'd like to do is explain what these are, because a lot of it is what I would call alphabet soup. First, just to give you an idea of some of the pharmaceutical negotiations that have gone on, you can see that the UK on the left-hand side has already negotiated to have an agreed supply, mostly with the Oxford vaccine, of about 5 doses per capita, but other vaccines as well. And you can see that the US is second in this potential expansion and negotiation, where if you look at the bottom of that chart, you can see that the low and middle income countries really, and I'll talk to you a little bit about how COVAX is negotiating for them, have really not agreed to any large amount of supply. And you can see on the right-hand side, just the 10 worst affected countries and their mortality, well, deaths per 100,000. And you can see that these do not correlate with the pharmaceutical negotiations that have been going on. May I have the next slide? So what is a potential global cooperative approach to the vaccine? Well, I was going to explain what I think is really the right way, and we can have Q&A about that, but that's the COVAX facility, which WHO, CEPI, and Gavi have put together. It's based in Geneva. 170 countries have bought into it, with 78 high-income countries, to try to collaborate to have an equitable access to the vaccine. What this COVAX facility does is it pools resources of countries and supports R&D, has a large portfolio, but the majority of the R&D support is going to 10 vaccines. It supports pricing principles consistent with benefit to the manufacturers, but also ensuring affordability to the participants. And there are two phases to this COVAX facility. One is whoever buys into it, and it really changes with your capability to buy into it. You get 20% coverage of your population. And phase two will be distribution to areas of highest risk. May I have the next slide? So it's a sort of insurance policy. And within COVAX, they have what we call the advanced market commitments. And these are funds that guarantee vaccine volume to manufacturers before licensing, somewhat incentive to get these manufacturers to produce vaccines to low-income countries. It purchases advanced doses when vaccine is licensed and WH pre-qualified, and it allocates supplies equitably and with a focus on epidemiology of the pandemic. The Gavi AMC and the COVAX is hoping to raise $2 billion. About $600 million have been raised so far. May I have the next slide? So I'm going to end with this, which I think is a very poignant quote from the director of the WHO, which we call him Dr. Tedros. And basically he says that excess demand and competition for supply is already creating vaccine nationalism and the risk of price gouging. This is the kind of market failure that only global solidarity, public sector investment, and engagement can solve. Thank you. I think we move on to Dr. Grant. And vaccine studies at Stanford.
Hello, hopefully people can hear me. So a couple of studies we're going to be running at Stanford. So go ahead, Kari, next slide. One study we're going to be starting pretty soon is the Ensemble study. That's the sponsor is Johnson & Johnson / Janssen. And as Kari mentioned, it's a replication incompetent Adeno26 vector. And it basically, within the core of that, it delivers encoding reagent for the SARS spike protein. One of the important things I think about it is it's been studied in a lot of different diseases already, and it's been shown to be effective for Ebola. So it's approved for that. It's originally actually was designed in Boston at Harvard for HIV, kind of the structure of it. So safety data, at least when those different viruses are being targeted, has been good so far. We've shown that a single Adeno26 vaccine, it produces a neutralizing antibody in response to SARS-CoV-2. The phase 1/2 data are under review at the FDA right now, and there's been no significant safety signal noted. But that's in a few thousand as opposed to the large phase 3, which will be looking at 30 to 60,000 participants globally at about 180 sites. We're going to be running our study over at the CTRU at Welch Road. There's minimal study visits. A lot of the stuff is going to be happening online. So you'll have an e-diary that you'll capture adverse effects. And if you do have symptoms concerning for COVID, there'll be a health officer that will go to your house or home health and can swab you and things like that. So most of it will occur off-site, but there's 3 study visits where you have blood draws, entry, and then about a month later, and then a year later as well. Based on how things are running, we hope to have our first participant on study in about a week and a half. We have our site initiation visit next week, and contracting is ongoing right now. We've contracted to enroll about 1,000 participants at our site. The marketing materials and a website are still in process with IRB. So if you're interested in participating, the study coordinator that's managing enrollment is Saran, and that's her email. And then Sumana is our research manager, who also you can email. And then after we get everything up and running, we'll give you contact in terms of, there's also gonna be a broad advertising campaign that's run through a sponsor to make sure we get target populations and high-risk populations on study. So next study. This study, sorry, it's not a, this is a study that's being run by Dr. Maldonado, and she'll provide more information later. And Kari introduced the vaccine. It's with an adjuvant. Someone asked about how different adjuvants, each company has its own kind of adjuvant that has proprietary material, but this is with the M1 adjuvant. The phase 1/2 data Kari had shown, you're going to get 2 vaccines for this study, day 0 and day 21. The adenoviruses vaccines are going to get one shot, and this algorithm shown to be safe and produce levels of antibody higher than that found in convalescent serum. And we're hoping that, as in all the studies, we're hoping this actually is going to protect against infection with coronavirus. The phase 3 study is going to be beginning mid-October, and there will be about 500 participants. And again, details to follow from Dr. Maldonado. Dr. Balke is kind of the right-hand woman of Dr. Maldonado, as I understand it. So she'll be the first person to contact in terms of getting enrolled in the study. One of the things that I think we can talk about, Kari, and maybe, you know, we talked about Operation Warp Speed, and that's so focused on efficacy. And I think one of this, the light, you know, the study stopping just overnight, you know, the safety with these, you know, the speed we're going is going to really have to be very important in terms of these. As we've overlapped a lot of the phases, we're going to be very careful to make sure these vaccines, as we roll them out, are going to be safe. That's all I have to say.
Thank you, Dr. Grant. It's really a pleasure for us to have you and include you here. We are very happy to be able to know that you are the PI of this study that will be the first ever COVID vaccine here in the Stanford area. So we hope people will contact you and contact the people that you've talked about in your slides. And thank you again. Dr. Grant is on faculty here, a professor in infectious diseases, as well as runs many clinical trials, including the vaccine trial with J&J that will be starting hopefully soon. Thank you for that update. I wanted to also talk to the audience about some resources that are available to you. These slides are available to you as well. These are links to the World Health Organization, candidates for vaccines. The New York Times has a wonderful vaccine tracker. And then we have a New England Journal information website, which is very helpful overall to think about coronavirus and to understand both medications and diagnostics as well as vaccines. And importantly, Stanford website, Jack Zhang, who is one of our wonderful information technology staff members, has developed a website with others called A Race to a Cure. And I would suggest everyone goes to this website. It's just very well done and overall presents a lot of data in a very understandable way. FDA also has more information on vaccines, and then there's a tracker by the FDA. And then lastly, because of the recent transverse myelitis case, I wanted to bring to your attention a good article on adenoviral vector immunogenicities for those people that are interested. Finally, as I spoke about earlier, being able to understand natural immunity and whether or not antibodies and T-cell responses will inevitably protect someone against reinfection, as well as to understand new targets and viral transmission rates, I wanted to point out that we do have a COVID long-term immunity study going on here at Stanford, thanks to cooperation of the Biobank with Drs. Blish, Andra Blumkamp, myself, Dr. O'Hara, Dr. Rogers, and Dr. Yang. There are many principal investigators involved in this study, and it is being run out of the Crown Clinic, started by our wonderful colleagues in medicine, as well as Dr. Ortandi, Dr. Barman, and Dr. Vaughn are currently working with us to be able to see patients. If anyone has any referrals or you're interested in the study, please let me know. We're very grateful for funding from different agencies, as well as being able to have these patients followed over the long term. And we'll hopefully follow them up to 4 years now with COVID, as well as those people that are in their households. I want to thank you, thank the audience, and thank the US FDA, Stanford Medicine, as well as the NIAID and the NHLBI. I'll stop there and we will answer questions. Thank you again.
Thank you so much, Kari, for a wonderful discussion, great panelists. We have a number of questions, and I think this one that's addressed clearly to Michelle, and that is, has the US bought into the COVAX facility for making vaccine available globally?
Yes, I'd be glad to answer that. Unfortunately, no. As part of this administration's denial to the WHO funding, they've also announced that they will not be buying into COVAX. Now, one thing that you should know is the WHO, that the US government does support CEPI and Gavi. So we're hoping that some of that funding will be channeled into COVAX. And as far as fair allocation of vaccines, there is a National Academy of Medicine task force now that is open for public comment if anybody wants to make comment to it about allocation of vaccines in the United States, equitable allocation. And that's being run by Helene Gale.
So can I ask a question of all the panelists, and that is, what is it about transverse myelitis that makes it emerge as a side effect historically for vaccines, and what other side effects can one expect from this really plethora of vaccines that are out there? It seems reasonable to think that we're going to see side effects. What will be the spectrum from history of what we can expect?
I think it's an excellent question, and I'll ask Dr. Grant and Dr. Berry also to speak on this, since they are infectious disease specialists. And as a pediatrician, I deal with vaccines all the time, and of course it's on the parents' minds as now as adults as well, with what are the potential side effects of any vaccine, and especially now that we're dealing with different platforms of vaccines that we haven't used before. We need to be very circumspect and cautious and show equipoise in the way that we develop these vaccines. But I think if you look at the typical adverse event profile of a vaccine, there are numerous amounts of inflammatory mediated side effects that occur rarely, but they can occur. And so we need to make sure those are documented. And like Dr. Grant mentioned, if we go too fast out of this, we could miss things, or we could inappropriately assign harm to a vaccine that it might not necessarily be associated with. So to make sure that we understand cause and effect, to make sure that we dive deep into any one of these symptoms like transverse myelitis, find out if it was an autoantibody, make sure that we can manage the patient, and then see whether or not we can modify a vaccine so that there's less side effects. I think these are all going to be critical. But Dr. Grant and Dr. Berry, what do you think?
I think the other thing that people talk about is enhancement. If you don't get a good antibody response, you can get a worse infection if you do get infected. So that's one of the concerns that gets brought up. So as Dr. Nadeau said, autoimmune stuff, enhancement, the things that the swine flu, we saw Guillain-Barré, so another autoimmune effect. Narcolepsy was seen with a vaccine for H1N1 during the 2009. These are all rare side effects. And I think it's going to be very hard in this 24/7 hour news cycle as things get rolled out for the public to sort between the wheat and the chaff, as Dr. Nadeau said. So I think it's really unprecedented in a lot of different ways. This epidemic, how fast things are moving, how fast the science is moving, how fast the news cycle is moving, and how politicized the environment is. So I think it's going to be a challenge for, I think, scientists and physicians to kind of provide their patients the right information and kind of avoid all the noise that we're hearing right now around in terms of safety. But I think it's important. We're putting out new platforms for vaccines, so it's going to be important to see if there's any difference in terms of what types of adverse effects we may see.
Several questions here about. I'm sorry. Go ahead.
Dr. Berry, did you have any comments on that?
There are a lot of questions.
Okay.
So several questions here about the target populations. given that there are so many phase 3 studies that will be out there, how do we compare them to each other? And also, is there sort of a universal agreement on who we should be testing? What is the role of prior antibody levels or exposure in the folks we're testing? And then what about concurrent flu vaccine administration? So several questions along that line.
I'll start with the latter, and then we'll move forward with your former question. It's an excellent question. Then I'll make sure Dr. Grant and Dr. Berry get to comment. This is really important since Dr. Grant is running the phase 3 for the Janssen product, the J&J product. So the CDC has come out, as well as the FDA, that for an otherwise healthy convalescent COVID-19 recovering patient, that they can receive the flu vaccine. And that's very important, is that all of us continue to get vaccinated with the guidelines, and that with the flu season coming, especially for those people that had COVID, they should also get the flu vaccine. And we're also trying to study that here to make sure that we understand how the patients with COVID had potential changes in their immune responses to potential vaccines like the flu vaccine. To answer your question about the initial phase 3 trials, a lot of them are about 30,000 patients each, 20,000 active, 10,000 placebo. Those are going in various sites around the country, but they're mostly in adults. But each one of them is slightly different in terms of their eligibility criteria, with whether or not someone who has antibodies is eligible. And I'll ask Dr. Grant that, because he probably knows a lot more details into what's going to be expected for a typical phase 3 study and eligibility criteria.
So for our study, I can say they're based on which site it is, they're going to do baseline antibodies based on, so in areas where there's thought to be a lot of infections, participants at that site are going to get antibodies performed. It's not an exclusion criteria, but they are checking antibodies. At our site, they're not going to run the antibodies, although we're going to collect plasma, and it can be done in retrospect. I think there's so many questions. I think I'll just answer that specific one.
Several interest about, Michelle, did you have an answer? Did you have a response to that?
Okay.
Go ahead.
No, you go ahead, because there are
several questions about anti-vaxxers, and we're dealing with a population who's inherently not accepting of vaccination, given, as an example, see the emergence of measles. So how do you factor that in? A vaccine that's partially effective, a population that isn't necessarily going to take it. How does one predict what that's going to do in America, for example?
Well, I think that we need to be very careful and show a lot of caution in being able to not be overzealous in our interpretation of results. I know we all, of course, want to be able to have a vaccine to reduce this pandemic and to get back to normal life, but we need to be extremely careful with not over-interpreting results, as well as potentially putting people at risk for safety. I do agree with Dr. Grant that, one bad case could really affect the public respect and integrity for our profession, as well as for research. And so we need to be very careful as to how we roll this out, make sure we know the safety in large populations and different ethnicities, make sure that we look at different age groups and different sensitive populations. So with that, the anti-vaxxers, it's a very difficult position to come from. Of course, vaccines are critical to help us and to make sure that we can have global protection against pandemics. Like Dr. Fauci said, we probably only need about 50% effectiveness to be able to mitigate the pandemic spread to some degree. We might not be able to get to 100%, which seems very sure, but importantly is to make sure that people that are anti-vaxxers and this current climate, that we base our facts on science, and that we do not base our facts on myths or on falsehoods. And so for us to make sure that we communicate the data well to the public and have them understand that all of this work has been based on science, and that the overwhelming evidence has been able to allow us to have strong facts about these vaccines and whether or not they're safe or effective.
Abe, can I make a comment on that?
Please.
In mid-September, we're having Heidi Larson from the London School of Tropical Medicine come speak here. She's the leading, really expert in vaccine confidence in what we're calling the misinfo epidemic of misinformation about vaccines. So the Center for Innovation and Global Health is sponsoring her, so stay tuned for that.
Wonderful. Michelle, could you further comment on the fact that the WHO has been hamstrung by in its efforts by the US government's pulling away from supporting it? But that aside, how effective has it been? How effective can it be? What is its strategy going forward? And if you were advising, which you probably are, what would you be telling Dr. Ted?
Well, I would say this is misinformation also, because I think the WHO has done a very excellent job in distributing PPE. Well, first of all, let me start. The WHO is not a perfect organization. What the problem is that its main funding comes from donor states. So that Dr. Tedros has to really manage between diplomacy and action. And if you look at what they've actually done, the COVID-19 response has been amazing. They have basically distributed PPE around the world. They've sent multiple experts. They've distributed the genome. They have not criticized member states, and you might take aback about that. But in their actions, they've been pretty amazing. What I would say to Dr. Tedros is that he really needs to get the WHO, and we all need to get the WHO depoliticized so that it's not dependent upon member state or donations, that it has its own independent funding.
Anyone think that we will really have a vaccine by November?
Does anyone want to take that? I think we have to be very careful in making sure that if we do a vaccine in November, it has to have been through phase 3 trials and it has to have met some very rigorous criteria. I think that would be hard to be able to have by November this early in the game. As you saw, many company CEOs are already saying they will not activate the EUA mechanism. They want to make sure they have solid phase 3 data in large trials to be able to make sure that their vaccine is effective and can move forward. Dr. Grant, do you want to talk about that, given the fact that you're starting something in September?
Right, I think it's each study, if you look at their protocol, has DSMB meetings based on number of accumulated infections. And so I think the Moderna study is, I think it's 54 number of total infections. In order for the DSMB to stop the study, you need to have a very, very strong effect at that sample size. And if we're thinking the vaccine efficacy isn't going to be, like Dr. Fauci said, 90%, I wouldn't expect they would stop the study with the first DSMB meeting. I've heard it may be occurring in October, but that's all rumor. You know, everyone's trying to capture, because DSMBs usually operate a little bit behind the scenes, but you know, we are seeing everyone's interested when is the first DSMB meeting. That's potentially when the study could be stopped if there was strong efficacy data. But I think it's been really nice, the pushback against the hurrying out of this vaccine. I think John Wooden, a famous basketball coach, said, "Hurry, don't rush." And I think that's what we're trying to do: be safe, but be expeditious.
So I'm going to put each of you on the spot and ask: would you enroll in a trial right now if it were offered to you? Do you feel you have enough data? Do you want to see more safety data?
I've signed up to be a volunteer to the NIH studies. I haven't been called yet.
Yeah, I'm hoping to enroll in Bonnie's study when it's, because you can't enroll your own, but when it starts in October.
Yeah, I think I would enroll to be of service and to be part of a clinical trial.
Yeah, I think that would probably be the caveat, that we'd all enroll, but as part of a clinical trial. Great. So we have a few minutes left. I want to ask the panelists if they have some sort of concluding thoughts before we thank you formally for doing such a great job in giving us really an exciting, uplifting update on what's coming. Who wants to go first?
Dr. Barry, do you want to go first
Sure.
on your thoughts about global distribution?
Yeah, I was
Yeah.
waiting for you, Kari. Yes, think to me, the most important thing is to vote November 3rd to try to get an administration that will support WHO. I think it's amazing that we're not supporting the WHO at this point, and that we need to support COVAX and global cooperation. There are many issues, existential issues, such as climate change, pollution, what we're seeing with these wildfires, that are threats, and pandemic diseases that are threats that need global governance, or what I've called shared governance or interdependent sovereignty. So yes, I think it's really important to vote November 3rd. That's my last thought.
Thank you.
Thank you. Dr. Grant, do you want to spend your last minute?
No, I think it's, I think one of the things is that people have so many questions, and all of them valid questions, you know, around how long it's going to last, how would it interact with other vaccines. And I think that's important why we do the science. Most of those questions are unanswered right now. So I hope and the other thing I think in terms of a lot of people are hesitant to get vaccines, and I think we should distinguish that, you know, vaccine hesitancy from people who are truly ignoring the information.
And Kari, can I add one other thing about equitable allocation? I think that needs to be first and foremost as we roll these vaccines out.
I think, as David Magnus said the other day, this COVID pandemic and the way that we're rolling out the vaccines is going to be an x-ray into the soul of our nation in terms of making sure that we're equitable and have people gain access to very important medicines as well as vaccines. I would only part with the fact that I hope today that we've shown the audience that there's hope and promise, that there are many different vaccines moving forward in the pipeline, and that inevitably there will be one that is safe and efficacious to the degree that we need it to provide some mitigation of the pandemic. So we are in a better place than we were months ago. And I truly believe as an optimist that we're going to be able to get there, but we have to be able to be cautious and circumspect and move forward with scientific data and careful analysis of that data.
I want to just remind the audience that we have many outstanding questions that we haven't gotten to, but we will try to be answering them and put them online. I think we're just incredibly blessed to not only be doing vaccine trials here, but to have such expertise both in the global health arena and in the arena of vaccines in general. Kari, thank you for putting together such an extraordinary panel and all the hard work you did, all of you did, curating the information. We're very grateful to you, and I can see us having an update in the future where we report back on where we are, you know, 6-7 months from now, when we have a lot more data. Thanks to our audience, and we'll see you all next week. And again, heartfelt thanks to our panelists.
Thanks, Abraham.
Thank you.
Thank you.